Okay, the recording is started. There's a couple of things I wanted to say. Again, you've heard me say it 1,000 times. Today is the kickoff of our end of course evaluations. You should have gotten an email in your Lake Hawk mail letting you know where you can click to go ahead and complete those. Please do that for me if you would. It's very important. I've mentioned to you a number of times about how it helps me make decisions about books, about how I present material in class. So, do put in the effort and fill those things out there. You can be done very quickly. If you're on campus today, I'll be hanging around in the science and math lobby with a bunch of computers, encouraging students to do their surveys as they kind of come through the lobby. And I'll have candy and little giveaways if you want to come by. And even if you've already filled out your survey, if you're on campus, come by and I'll throw you a piece of candy or something. There's also going to be other little things like pens and things, just little doodads. Anyway, so please do complete those and get those finished, as soon as you can. Oh, yes, the lab practical next week. I'm going to do the same thing we did before. I'm not going to have a lecture before the lab practical, so the lab practical will start at 11. I'll send out an announcement in Blackboard just so everybody knows. So on Tuesday with your lab practical, no lecture. It's candy. I mean, that's kind of payment, right, Penny? I mean, we got little doodads. Now, I always tell students that I like reading the reviews and they talk about, they enjoyed my class. I like that too. But I also like when people tell me things about that they liked or that worked well for them and didn't work well for them. So, I really what I want mostly on the reviews is honesty, because I'm trying to adapt and change my course as I, as life changes and as the student body population changes. I mean, you all grew up in a totally different era with different kinds of access to different pieces of equipment that even some of my first students, when I started teaching, what year is this? Way too long ago. 30 years almost? Maybe this is 30 years. I started teaching in 90. No, not quite. 27 years. All right, anybody got any questions on any of that sort of stuff before we get started on chapter 15? I am going to probably end a little bit early today. I've got something that I need to do, which I just explained to you, so I need to get that all set up. But I do want to get through most, if not, we may not get through all of 15, but I want to get well into 15. We've only got this and then 16 to go, so we don't have a whole lot. Seems like we have a whole nother chapter in 16, but we're only doing a bit of 16, so that's going to be a relatively short lecture. This is actually the last, all right, Penny, sure. This is actually the last lecture that's going to be relatively long. So what we're going to talk about here in chapter 15, again, goes back to the very beginning of the semester when we talked about the shape of proteins. We spent all that time talking about it, and I mentioned to you at the time, I said, we're going to keep talking about this, and at the end of the semester, we're going to kind of do a full loop and come back and talk about it again. If you remember, what a protein is a sequence of amino acids, right? And you studied the amino acids in lab, you studied for them on the lab practicals, or the functional groups you studied in lab and on the lab practicals. But we talked about amino acids in lecture. We talked about there's 20 different amino acids, phenylalanine, leucine, all those things that we introduced back in, I don't know, was it chapter 5? And we said the way you make a protein is you link amino acids in a certain order. And because of the side groups, and because of the side groups that those amino acids have, those amino acids are going to cause that chain of amino acids, that protein, to fold into a particular shape. And what's the most important thing about a protein? What's the most important thing about a protein? Absolutely, right? It's shape. So you got to get the right amino acids in the right order. What you're looking at here is a decoder table. It's really kind of neat. It reminds me a lot of the little decoder rings we used to have as kids in school, where like you used to give them in cereal boxes. I could write a message to my friend in a code. As long as he had the decoder ring that I did, he could break the code. So this is kind of what that is. And what you're looking at here are bases. These are RNA bases. There's uracil and cytosine and adenine and guanine. That's how you know it's RNA. And what you see down below, the sequence of 3 letters is a code, or more specifically, we actually call it a codon. And you can see right away that a sequence of those 3 bases codes, or is a codon, for a particular amino acid. That's the 3-letter abbreviation, like up here, UUU. Wait, start easy, right? UUU. UUU, those 3 bases, that codes for the amino acid phenylalanine. UCU right next to it is serine. AAA is lysine. You notice that there are a few of them that I've colored red. Those are what we call stop codons. So we'll talk a little more about how all these work in just a few slides. But we're going to figure out how your cell takes this sequence of letters and turns it into a sequence of amino acids. All right, so what we're going to be looking at is this whole process known as the central dogma. And the central dogma is an accepted way that things occur, all right? So the central dogma, the idea is the DNA is your code, right? And the DNA is the archive of your code. So in other words, your DNA molecule really doesn't interact directly with the production of proteins. Okay, what I mean by that is the code for that sequence of amino acids is in the DNA, but the DNA is like the archival library copy. What we do is we make a piece of RNA, so that comes back to that other nucleic acid, and there are different types of RNA. We'll get into those in just a minute. But you make a piece of RNA that matches the code on the DNA, right? Remember, Chargaff's rule said adenine always binds to thymine, guanine to cytosine. The only thing you have to modify with that is, since we're talking about RNA, it's not going to be a thymine, right? It's going to be a uracil. So that sequence of letters that's in the DNA, A, G, T, whatever that is, that code, your genes, are used to create a piece of RNA, specifically a piece of RNA that we call messenger RNA. It's got a little M in front of it. And that piece of messenger RNA is a copy of the code. It's a copy of the DNA code. See, your DNA code is very important, obviously. So you're not going to want to interact with it a whole lot. You're going to want to use it as, like I said, sort of the reference code, right? And the RNA is what's going to actually deliver the code out to the ribosomes. You remember the ribosomes are where proteins are made, right? So that last step in that central dogma, where it goes from RNA to protein, is now that RNA, that messenger RNA, you can even see in the pictures down below too, is delivered to a protein, and a ribosome, which of course is partially a protein, and the ribosome reads that code on the messenger RNA and turns that into a sequence of amino acids, or have you seen the bacterial cell, a polypeptide, which is another name for a protein. All right, so you can see, like in the bacterial cell, this is a simple process. First, what we're going to do is we're going to copy the code, so we're going to undergo something called transcription. Transcription makes a piece of messenger RNA, like I said. All right, now we don't turn the DNA into messenger RNA. The DNA is just used as the reference code to make the messenger RNA. That message, that code, then goes out to the ribosomes, where it's then translated. Each 3 sets of letters on the messenger RNA, like I showed you in the previous slide, codes for a particular amino acid. Now, it's relatively simple in bacterial cells. It's a transcription translation kind of thing. There's no nucleus, of course, because they don't have a nucleus. Little different in eukaryotic cells, though. For one thing, the messenger RNA, when it's produced during transcription, is actually not quite finished yet. It's actually, in this case, called pre-messenger RNA, and it has to be processed. We'll talk a little more about why that process is necessary. And then it's got to be exported out of the nucleus. But then the same kind of thing happens, right? Translation out on the ribosome. So the messenger RNA is transcribed, it's processed, leaves the nucleus, finds a ribosome where it's then translated, right? So if I understand kind of so the rest of this chapter that we're going to go over, the rest of this lecture, is basically going to look at the details about how all this happens. But this is, if you will, the forest of the trees, right? This is the basic process. We're going to talk about how do we take that sequence of letters on the DNA and turn it into a sequence of amino acids. So maybe something like this, right? So there's a DNA molecule. One of those strands Remember, DNA is anti-parallel. There's two strands, is known as the template strand, and that's the strand that's going to be used in transcription. The other side, like for example, this here is the template, ACC, AAA, CCG, AGT. The other side of the DNA molecule is there really for protection, keeping the molecule together, keeping the code faithful, limiting the amount of changes that can occur to the DNA code. So there's only one side that's read. The other side is kind of like the packing material, again, kind of thing that I've talked about. So during transcription, that template strand of DNA is going to be read and turned into, not turned into, but that code is going to be used to make a piece of messenger RNA. And again, it's very simple to figure out what the codons or the bases are going to be in the messenger RNA. It's Chargaff's rule. So the adenine in the template strand is going to be a uracil, not a thymine. Remember, there's no thymine in RNA. The two cytosines are going to be the two guanines. Then the three adenines will be the three uracils, and so on and so forth. Out in the ribosome then, like I said with that first slide, those three bases are read three at a time. That's called a codon. So the ribosome reads UGG, and it knows in its ribosome machinery that that's talking about the amino acid, what is that, tryptophan? All right. UUU, the amino acid phenylalanine. GGC, glycine. UCA, serine. So that's where folks, this is the primary level of protein sequence, protein structure, isn't it? This is the primary level. The primary level of protein structure is the order of the amino acids. And as I said to you back then, when we were talking about that, if you don't get the order of the amino acids right, you potentially affect the shape of that protein. All right? So this is where that code comes from. This is the recipe. And as I said, we have the code table for any set of codons. We've broken the code, if you will, to tell us what amino acid that is. Here's the code table. Now, do not memorize these code tables. I don't have this code table memorized. I have some codons memorized only because I've been talking about them forever. But if on the exam I want you to do a transcription translation exercise, so I give you a piece of DNA with a sequence of letters, and I ask you to transcribe that, make a piece of messenger RNA, and then act as if you're the ribosome, and then assemble the amino acids in the sequence. I'll give you a code table, though. I'm not going to expect you to memorize that, but I do need you to know how to read the code table. The code table is made up of the left-hand side, which is the first base in the codon, the second base across the top, and the third base is down the right-hand side. So if I wanted to know what codon is going to, or what amino acid is going to be coded for by, let's say, CAC. All right, so that's on the messenger RNA. First base is a C. Second base is an A. Third base is a C. The intersection of all those gives me this amino acid. That's histidine. And again, don't worry about memorizing the name of the amino acids. Makes sense, right? Pretty easy. Now you guys try it. What amino acid is coded for by the codon AGU? AGU. Right, serine. What amino acid is coded for by the codon AGC? Same one. Notice the codons CGU, these four right here is what I'm talking about. Notice they all code for the same amino acid, arginine. Mother Nature realizes that things aren't perfect during transcription and translation, so she builds in a little bit of a fudge factor. We call the last base in that codon many times, depending on the amino acid in the codon, we call it a wobbly base. In other words, it's kind of like a wild card. So for example, with arginine, to get an arginine amino acid, all you really need to have right is the first two bases, right? All you need is CG. If it's a U, C, A, or G, anything, it's going to be the same amino acid. With the example I gave you with serine before, AGU as well as AGC will give you the same amino acid. So it accommodates those slight little changes that might occur in the DNA. So once again, Mother Nature realizing that things sometimes are a little bit out of whack, she builds in, just like I said, a little fudge factor or a little safety net, if you want to think about it. For the most part, as long as you get the first two letters right, as long as those are correct in the codon, you'll get the right amino acid. Now, that isn't always the case. Like if you look at, for example, AAC and AAA, and as I said, it varies from the codon and the amino acid. Those code for two different amino acids. Okay? Wobbly bases, Mother Nature's way of making sure that you're not getting penalized for every single little mutation. There's a couple of special codons. AUG, look that one up for me. AUG. Yeah, so there's no amino acid called START, but there is an amino acid known as methionine. That's the START codon. So a sequence of amino acids, a protein, is going to start with the START codon. The three in the upper right, UAA, UAG, and UGA, there obviously is no amino acid known as STOP, so this is the STOP codon. What this means is on a piece of messenger RNA, on one piece of messenger RNA, so let me draw a line to indicate this is my piece of messenger RNA. I can have several start and stop codons. So a start codon, stop codon, then a start codon, and a stop codon. What I can do is I can code for, in this case, three different sequence of amino acids, three different proteins. So the start and stop codons are kind of like, when your song switches over on a CD back in the days when they used to have that, right? It's when the track switches over. So it makes it very efficient, doesn't it? Make a big piece of messenger RNA, separated between a bunch of start and stop codons, send that out to the ribosome, and you're cranking out tons and tons of proteins. Sometimes proteins are very complex, if you remember, from the quaternary level, and you need more than one, right? At the quaternary level, more than one protein subunit. So each of those protein subunits could be coded for, and then they can bond together and form that fourth level protein. It's very efficient. All right. Let's kind of now drill down a little bit further and see how the machinery works. We're going to start up here at the top. There's a piece of DNA. The 5' and the 3' once again is reminding you that it's anti-parallel. When a gene is going to be coded, or when a gene is going to be transcribed, we have an enzyme that does that. You see, it looks like a big guitar pick, the one thing at the bottom, the RNA polymerase II. You remember there was an enzyme known as DNA polymerase when we talked about DNA replication, and DNA polymerase, enzymes sometimes tell you what they do, creates polymers of DNA. RNA polymerase creates polymers of RNA. All right, not DNA, of RNA. That's the little red that you see in the picture. But how does the RNA polymerase II know where to start? All right, there is the start codon, but that's for the first amino acid. I'm talking about as the RNA polymerase is moving down the DNA, how does it know when it's getting ready to read a gene? Well, it looks for something known as a Tata box. And the reason why a Tata box is called that is you can see right away it's TA, it's a bunch of T's and A's together. So the DNA polymerase is helped, first of all, by these things called transcription factors. They bind to the Tata box, and then the transcription factors promote the binding of the RNA polymerase II to the DNA. All right, so it's the transcription factors binding to the Tata box, which then, and it's all a chemical kind of thing, which then promotes or encourages or makes it more probable that the RNA polymerase II is going to bind where those transcription factors are, and as soon as it does, then we're off to the races, right? So we start making that piece of RNA, that messenger RNA, that RNA transcript in the picture, we start making that from that point forward, all right? So that RNA polymerase is going to move from left to right, reading down the template strand. And you'll notice it's reading the template strand the same way you have to read it when you make a copy of DNA, from the 3' to the 5' end. So it's going to read down that template strand, and it's just going to copy the. It's just going to, it's going to make a copy of the RNA, or make a piece of the RNA based on the code that's in the DNA, which is basically what I just said right here, right? So there's the promoter sequence, the transcription unit, that's the gene. So the RNA reads down the template strand, and as it does, it cranks out that piece of messenger RNA, the part that's in the red. And it does this, like I said, A's and T's and G's and C's. Here's a little bigger picture. So everywhere there was a thymine in the template strand, the RNA polymerase is going to catalyze a reaction in which a free RNA nucleotide, see those RNA nucleotides, they exist there in the nucleus of the cell. They're like the letters on a typewriter. They're ready to be used to be put into a sequence. So RNA polymerase is going to bind an adenine to the thymine, or uracil to the adenine, cytosines to the guanine, and go on and on and on. Now notice it's only reading one side, right? It's only reading the template strand, and you always remember the template strand is the strand that starts from the 3' and ends with the 5'. So the direction of transcription is moving this way down that template strand, which is the ones I just circled, the 3 to the 5'. In eukaryotes, they do this at about a rate of about 40 nucleotides a second. So I guess that's pretty fast. Right, RNA polymerase moving down that piece of template strand, about 40 nucleotides a second. And Mother Nature, again, being very efficient, RNA polymerase can be transcribed, or RNA polymerase can be created out of transcription throughout several genes simultaneously. So there's the start and stop codon thing that I was talking about. But now what you're looking at here are chromosomal puffs. And what chromosomal puffs are, let me go back to the previous picture, is this area that's puffed open, right? So the DNA is unzipped and it's spread out like that. That's what chromosomal puffs are. So whenever you see like there and there and there and all along in here, here's the DNA, by the way. All those little circles are puffs, and they're places where the DNA is opened up and the RNA polymerase is reading and making pieces of messenger RNA. Aren't those cool pictures? Isn't that neat? You can see that thing in the upper right even looks like a chromosome, right, with the banding. Okay, let's talk a little bit now about some of the differences between prokaryotic and eukaryotic cells. And first of all, let's talk about eukaryotic cells, and then I'll introduce you what's big difference about the prokaryotic. So this is the part that I talked about earlier where the messenger RNA has to be processed. So in prokaryotic cells, there's no messenger RNA processing at all, only in eukaryotic cells. The first thing that's going to happen is we're going to get that piece of messenger RNA ready to leave the nucleus. And so that means we're going to add a couple things to the end of the molecule. We're going to put a 5' cap, so there's just going to be a series of bases, and we're going to put a poly A tail. Now the poly A tail is exactly what it looks like in the picture there. It's a bunch of A's, adding, just a bunch of them at the end of that. Those two things, the cap on one end and the poly A tail on the other, are going to allow or facilitate, make it easier for the messenger RNA to leave the nucleus and go out to the cytoplasm. It's also going to protect the messenger RNA from enzymes that would want to destroy it. See, these ends, remember we talked about telomeres with the DNA last time? So the poly A tail and the 5' cap, they're kind of like the telomeres on the messenger RNA. They're sort of like the protecting cap at the end. Now, why would these hydrolytic enzymes want to destroy the messenger RNA? Well, these hydrolytic enzymes, the RNA and DNA is one of the way that viruses, well, it's the way that viruses attack host cells. So you've got enzymes in your cells that would get rid of pieces of RNA, not knowing that necessarily this is a piece of messenger RNA that's actually you're trying to use to make a protein, thinking maybe it was a piece of viral RNA. So you have things that protect you from that RNA, but when you're making a piece of messenger RNA, you kind of put these caps on it so that we protect the ends of the molecule. And you notice that there's also an untranslated region at the ends as well. All that is protection. The exons down below, we'll talk in just a second what are introns and exons, but the exons are the genes, okay? The last thing that putting these caps on is going to help this thing attach to a ribosome. All right, now, messenger RNA processing, as I said, does not happen in prokaryotic cells. The reason why is because of the lack of introns. Introns are the part of your DNA that are not read. Remember I said that parts of your DNA have no discernible function. I mean, we used to call it junk DNA, and I think I told you at one time we used to say 95% of your DNA was basically junk DNA. It's not quite the case. A lot of that junk, there still is junk DNA, but a lot of that junk DNA has other functions that we found out as we've done more research. But the introns are the part, it's the filler part in the DNA. DNA makes copies of itself only because it wants to make a copy of itself. The fact that the genes are there is just something that we use. We use the chemistry of that. Now, prokaryotic cells don't have introns, so they don't need to go through this process. But during eukaryotic cell messenger RNA modification, while we're putting the caps on, we splice out the introns. As a matter of fact, it's something called a spliceosome. That's the big purple thing that you see there, which are small little pieces of RNA. And what they do is they go in. Now, the exon, this is so cool, man. Check this out. The exon is the red part. All right, the intron is the light red part. So we want to get rid of it. What it does is it takes the two exon pieces and it brings them together and it forms a loop. You see that loop of the intron down there? And then once those two pieces come together, the spliceosome enzyme, which is made up of a lot of these small little RNA pieces, chops out that intron and sticks the two exons together. Isn't that kind of neat? You think about that. I mean, it's just amazing, the machinery. Now, as I said in red, this is not necessary in those members of the domain bacteria. Now, three domains, archaea, bacteria, and eukarya. This is not necessary because members in that domain don't have introns. Everything they have in their DNA is exons. Everything is red DNA. Okay? So we put the caps on, right? That's going to protect it from those enzymes. It's going to help it bind to a ribosome. It's going to help it get out of the nucleus. We cut out the introns if we're talking about the messenger RNA in eukaryotic cells. And then that piece of messenger RNA leaves the nucleus of the cell and goes out and it finds a ribosome. And in the ribosome, this is basically what's happening. Translation. In the ribosome, that big brown thing is the ribosome. In the ribosome, there are three binding sites. They're little indentations that you can see there. Now, we'll talk about what all these pieces are. If you're wondering what that little green thing is, we'll talk about that in just a moment. But in those binding sites, yeah, they do. They do kind of look like socks if you had three toes. In those binding sites in the messenger RNAs where all of this activity is happening, right? This is where the polypeptide or the amino acid sequence is being assembled, is being put together. So the ribosome is reading those codons, and with those codons, it's assembling the a protein, the amino acids into a protein in the right order. Now, the green things that Emily said kind of look like socks. That's another piece of RNA. There are three types of RNA. Messenger RNA, we've already introduced you to. The other type is known as ribosomal RNA, which is abbreviated that way. And ribosomal RNA, all I'm going to say about it is it helps the cell make a ribosome. So we're not going to really talk about it much anymore. The other type is the one you see here with the T. This is known as transfer RNA. Those little green things are transfer RNA. And what they do is they transfer amino acids to the ribosome based upon the code that's on the messenger RNA. So transfer RNA molecules, you can see this one on the right-hand side, is bringing into the ribosome, so we're kind of catching this, halfway through the process, if you will, is bringing in the amino acid glycine. And that transfer RNA molecule is going to be able to bind into that binding site on the right-hand side because the codon and the anticodon match. Look on that piece of transfer RNA with the glycine, and then look at the codon that's on the messenger RNA. The code on the messenger RNA is GGC. The anticodon in the transfer RNA is the complement, CCG. You see that? Remember what I said before, these things can't think. Enzymes and substrates don't get together because they know they're supposed to get together. They get together because they fit. All these chemical reactions are this incredible, dance of life that we call it here. All are governed by whether things fit together, whether chemical reactions occur the way they do. So codons and anticodons match simply because they're the complementary of the base. right? The G has to have the C on the other side, and an A has to have a U because we're talking about RNA here. So if you think about it for just a second, the anticodon sequence that's on the transfer RNA is the same sequence of bases, U for T being replaced, that's in the DNA, right? Because the messenger RNA is the opposite of the template strand, and the anticodon is the opposite of that. So the anticodon is what's on the DNA, again, switching the T for the U. Now you notice there are 3 binding sites. On the left-hand side is a piece of transfer RNA that is empty. At the upper end, that little thing that looks like a wick is known as an amino acid attachment site. So that transfer RNA molecule on the left is the one that brought in this amino acid, tryptophan. So we've got, what, 1, 2, 3, 4, 5, 6, 7 amino acids in this polypeptide. So we're in the process. We've caught it halfway through. Phenylalanine has just been brought in by that transfer RNA in the middle, and glycine is now being ready to be brought in. And as soon as the codon and the anticodon bind, glycine will be added to the end of the chain. So it'll go tryptophan, phenylalanine, glycine, on and on and on until it reaches a stop codon. We'll talk about what it does when it reaches a stop codon. Obviously, it stops making the protein, but it's a little more than that. Now, what you're looking at in the background, out in the cytoplasm of the cell, all those little purple things, those are all amino acids. Those are amino acids that are in your cell. Those are amino acids that you consumed. Those are that you ate in proteins. Those are the source, the building blocks, right? And then there are transfer RNA molecules out there. Some of them have an amino acid attached to them. Some are empty, looking for an amino acid to hook up to. Now, if you look at this for a second, there's one thing that kind of maybe puzzles you. How does the transfer RNA molecule, I mean, we know, or we just talked about how the transfer RNA molecule matches up with the codon and the anticodon. That's bases fitting together. But how does the transfer RNA molecule with a particular anticodon know, again, in quotes, that it has the right amino acid attached at the other end? Right? I mean, the codon and the anticodon thing is easy enough, but how does that transfer RNA molecule in the picture that's bringing in glycine, how does it know it has a glycine? Because remember, if the transfer RNA molecule isn't specific for the amino acid, then it makes no sense to have anticodons and codons matching up. All right, we'll talk about that in actually just a couple of slides, but let me show you a little bigger picture of transfer RNA. These are 3 images of the same thing. You'll notice the suck one that Emily pointed out is the one on the right. Take the one on the left. Transfer RNA molecule reminds me of a subdivision with 3 cul-de-sacs. That's what it looks like. And you'll recognize the letters in there. Those are the letters that occur, the adenines and the guanines and the cytosines. The parts that we're going to be most concerned about is up here at the top. This is the amino acid attachment site. And then down here at the bottom, these 3 bases that are at the very tip of this last cul-de-sac, those are the anticodon. All right, so let's do a little exercise. That's the anticodon. What would the codon be for this particular transfer RNA molecule? What codon would it bind to? What would be the sequence of letters? Thank you, Courtney. Absolutely. And what would that sequence be on the DNA? There you go. Oh, I'm sorry. No, on the DNA, it's going to be the same. Excuse me, AAG. All right, Courtney, go back, and Victoria, go back to what you told me what the codon was. The codon is read off the DNA, so UUC is only going to be read in messenger RNA if the DNA says AAG. Remember, the anticodon and the DNA are the same. Sorry, I said yes to begin with. Hopefully I did confuse you. So it's the opposite of the opposite. Got it? Okay, good. Anybody else? We all good on that? So let's answer this question. How does a particular transfer RNA molecule pick up the right amino acid? Well, we've talked about specificity of things fitting together a number of times this semester, especially when we talk about enzymes, right? We talked about enzymes are specific for a substrate because their active site is shaped that way. the reason why a substrate binds to a particular enzyme. Now, inhibitors all aside, that stuff that we talked about, the reason why a substrate binds to a particular enzyme is because they fit together. So if you think about it, the way to make sure that the right amino acid is binding to the right transfer RNA molecule might involve an enzyme. As a matter of fact, it does. For example, the amino acid tyrosine. So there's going to be one of these enzymes for each one. The amino acid tyrosine is attached to the proper transfer RNA molecule based upon its anticodon by an enzyme. See that enzyme, tyrosyl tRNA synthase. Right, this big tongue twister right there. So each amino acid is going to have an enzyme that attaches it to the proper transfer RNA molecule simply because they bind to the active site. So it's the same answer we've seen over and over again, right? So this takes a little bit of energy. Matter of fact, it takes two cleaves of that phosphate. So we're going ATP all the way to AMP to have this done. So there's an energy involved in all this, and that's what I told you before about building proteins. They're expensive molecules. That's why the shape is so important. That's why we spend so much time making sure that we get the right shape. All right, so that answers the questions. How do we get the right amino acid on the right transfer RNA molecule? Based on what's on that transfer RNA molecule's anticodon, it's going to bind to a particular enzyme. Based on whatever amino acid, it's going to bind to a particular enzyme, and that enzyme is going to stick those two things together with a bit of energy, and we're off to the races. We get a loaded transfer RNA molecule. Make sense? The generic name, by the way, for this enzyme is amino acyl tRNA synthase. So we're talking about any amino acid, but again, each amino acid is going to have a specific enzyme that matches these two things together. Okay. Let me talk about what those three sites are. EPA. Well, the one on the left-hand side is easy enough. That's the exit site. That's the, if you look at the picture on the right, that's the transfer RNA molecule that's just dumped off its amino acid. That's where it exits, it leaves. The part in the middle is known as the peptidyl tRNA binding site. The peptide bond, remember that's a bond between amino acids. It's in that site in which the new amino acid is going to be attached to the chain. So in the picture on the right, the transfer RNA molecule on the right, this amino acid right here, when this transfer RNA molecule is in the middle site, the P site, then that little amino acid, that one by itself, is going to be attached to the end of that growing chain of amino acids. Now, it's not the transfer RNA molecule that moves from site to site. It's actually the ribosome. So the ribosome jumps to the right every time a codon is read. The transfer RNA molecules stay where they are. So if you're looking at that picture on the right, when that ribosome makes its next jump, the transfer RNA molecule that's in the middle will be in the E site and it will exit. The transfer RNA molecule that's on the right, which is in the amino acetyl tRNA binding site, right, where that enzyme we just talked about, that's going to move into the middle. And then the A site, which was just vacated, is going to wait for the next transfer RNA molecule to come in with the right codon. And so that ribosome is going to jump a codon down the transfer RNA molecules. And it's going to keep adding an amino acid based on the code. So it's going to start cranking those out. Now, this all starts at a start codon. Again, Mother Nature being this classic about efficiency. Ribosomes aren't sitting around empty waiting for a piece of messenger RNA. There aren't factories of ribosomes just kind of idly standing by waiting for a piece of messenger RNA to come by and to be translated into a protein. Ribosomes are actually unassembled until needed. That's why you see in the picture here, you see the brown part on the left-hand side and then the right-hand side, that's the ribosome. Ribosomes are made up of a small and a large ribosomal subunit. So those two pieces are apart until we're ready to start reading out a piece of amino acid or a piece of messenger RNA to make a sequence of amino acids. So the messenger RNA, the small subunit, there's a spot on the messenger RNA where it binds to the messenger RNA molecule, the mRNA binding site. The first transfer RNA molecule is known as the initiator because on the other end of it is a methionine. Remember, that's the first amino acid. And the start codon on the messenger RNA matches up very nicely with the anticodon you see in the inset there of the transfer RNA molecule. So the small ribosomal subunit is there. The messenger RNA clicks in to the binding site. That first piece of initiator, that first piece of tRNA, the initiator, one with the start codon and the methionine bind. Energy is then used. This is another endergonic reaction. That's why I said, again, these things are expensive to make. This is not ATP, obviously. This is GTP, which is, you saw in the Krebs cycle. Citric acid cycle. Wait, Krebs cycle. Yes, same thing. GTP was made and then ATP was made. But nonetheless, that energy is going to be used to put the large ribosomal subunit, attach it together, and then we're off and running. You see, the first piece of transfer RNA molecule starts in that middle site, the P site. So what we're waiting for now, what will happen next, is a piece of transfer RNA molecule, or a piece of transfer RNA, or a transfer RNA molecule. I'm saying too many words, I don't know why. With the right anticodon will match with that codon. And we're off to the races. Around and around and around, using energy each time. And the ribosome is jumping to the right, moving down the piece of messenger RNA. The transfer RNA molecules, this elongation stage that we're talking about here, they're going to stay where they are. And every time the ribosome moves to the right, whatever was in the P site is going to be in the E site. It's going to exit, like you see over there on the left-hand side. And whatever was in the A site is going to jump into the P site. And then, starting back up at the top, another piece of another transfer RNA molecule is going to bind. So we go around and around and around, and we keep doing this, reading those bases 3 at a time. Remember what we said with DNA about a frame shift mutation? How the reason why frame shift mutations can be potentially damaging is because an extra base is stuck in there, or one is missing. The ribosome only knows to read the codons 3 at a time, remember? It doesn't know if that base is missing. So if you get a frame shift mutation, as you go through the elongation stage, everything's going to be screwed up, because that one base that's either not supposed to be there or is missing. Any questions on this picture? Does everybody get an idea of how that works? Sort of a visualization of how that's all put together? It's really kind of funny because you look between the step number 2 at the bottom. For a second, the amino acid is attached to the polypeptide chain in the A site. Now, that's a little bit misleading because what's happening is the ribosome is translocating at the same time. So this little red arrow where it says peptide bond formation and this ribosome translocation that's occurring is all kind of occurring at the same time. So it's like a jump, an attachment. So it doesn't really stay in the A site, it's moved from there. Yeah. And this is going to keep going on. The ribosome is going to keep jumping down that piece of messenger RNA until, guess what? A stop codon. A stop codon. Now, there is no transfer RNA that binds to the stop codons. There are 3 of them, UAG and UAA, right? But there is something called a release factor. It's just another molecule. The release factor is not a piece of transfer RNA, but it binds to the stop codons, and when it does, that signals everybody to go, we're done. The last piece of transfer RNA molecule exits. The protein that was being made, the free polypeptide, exits or leaves, is disconnected and begins folding, by the way. Begins alpha helices and beta pleated sheets and folding back on itself and doing all that stuff that we talked about, right? All that's going to happen there. The ribosome is going to fall apart because it doesn't need to be used anymore. The release factor is going to hang around waiting for another stop codon to appear. And then the messenger RNA, at the end, what's going to happen in that, do you think? What's going to happen in that piece of messenger RNA? I mean, we're done with it. We've read the code. Yeah, it falls apart, right? It falls apart, and the bases, the adenines and the uracil, get recycled back into the nucleus so they can be used in transcription again. I tell my students, it's kind of like if you sit down at a keyboard and you type out. Once you use the letters, they're not gone forever. You can use them again. It's kind of like a buddy of mine used to always say in high school. He wasn't a big reader. He says, I'll just read the dictionary. It's got all the words in it. So if I read the dictionary, I've read all the books in the world, at least in that language. right? So that piece of messenger RNA is a valuable molecule. It's got bases on it. Those letters can be used again to make another piece of messenger RNA, right? How neat. By the way, it's kind of a curiosity, the first hereditary molecule on Earth, the first molecule we think that had the ability to have heredity, the DNA, was RNA. We think RNA actually came, or we're pretty sure anyway, that RNA evolved, if you will, as a molecule first before DNA. DNA became the archival information on the RNA. See, remember, like if you look at this, RNA's got the code, right? RNA is what activates the protein. It's not the DNA. That's what I mean about the DNA is not directly involved in the construction of the amino acid chain. It has the code. That's all it's got. But once that code is read, it's the messenger RNA that goes out to the ribosome and gets all this activated and gets all this working. I think we're actually going to finish the chapter today, and we are going to finish a little bit early, which is perfect. All right, so let's take a look at the last couple slides. This is kind of cool. In prokaryotic cells, again, going back to this idea of Mother Nature being so doggone efficient, if you look up at the top, the actual photograph, you see the thin line, that's the DNA, and the RNA polymerase is reading down the DNA. Now, if you jump down below, you can kind of see the red line is the RNA polymerase. And as that RNA polymerase is reading down the DNA, making the messenger RNA, ribosomes are attaching to that piece of messenger RNA and cranking out proteins. So it's all very efficient. And if you notice, you can have more than one RNA polymerase, more than one ribosome moving down a piece of DNA, more than one ribosome attached to a piece of messenger RNA. You're cranking out some serious amount of proteins here, aren't you? That's what we mean up above, the polyribosome. That collection of ribosomes along that piece of messenger RNA is a polyribosome. Now, I wrote in red, this only happens in prokaryotes. You only get this simultaneous translation, right? That's what the ribosome is doing, is translation, and transcription, which is what the RNA polymerase is doing in prokaryotes. Can anybody tell me why this can only happen in prokaryotes? That this would not be able to occur this way in eukaryotes? No. Think about what's happening here. We're transcribing and we're immediately translating. No, it doesn't have to do with that. Again, think about what I said. We're transcribing, immediately we're translating. Why can that only happen in prokaryotes and that can't happen in eukaryotes? Why could we not get this polyribosome arrangement like this? Or this? There you go, Victoria. Yes, absolutely. Thank you. Eukaryotic cells, remember, this is eukaryotes. You have to process that messenger RNA first, right? You got to put those tails on it. You got to get rid of those introns. Now, introns and exons, the words can confuse you as to which part is left in and which part is left out, because to me, exons almost sounds like it's X'd out. But what the words literally mean, exons are the expressed parts, right? The parts that are actually read. And introns are the intervals between the expressed parts. So remember it that way, you won't get it confused. But yes, in eukaryotes, we have to cut out the introns, we have to put the caps on there, and then out to a ribosome. So we wouldn't be able to do simultaneous transcription, which is what the RNA polymerase is doing, and then have a ribosome attached to it. That piece of messenger RNA that's coming out of that RNA polymerase in a eukaryotic cell, it's not done yet. It's not processed. Remember, prokaryotic cells, especially those in the domain bacteria, it's a little different in the domain archaea, the other domain that's out there, the other prokaryotic domain. But in the domain bacteria, they don't have those introns. So you can do this. You can't do that in eukaryotes. Eukaryotes, you got to process all that stuff. Okay? Folks, that's it for the chapter. I'm done for today. If you have any questions, if not, please, once again, fill out your course evaluations. Get those done now before you get busy when the next couple of weeks before the semester ends. It's very important. I'm looking forward to reading them from all of you and getting some good ideas about what to do next semester. I know that doesn't benefit you, but we're kind of all in this boat together, right? We're all supposed to be helping each other out. Other than that, have a good weekend, and I will see you, remember, Tuesday, and I will send out the announcement that the lab practical will start at 11 and there won't be the lecture. I'm going to stop the recording now.